This video will be on Chapter 9, Bacterial Genetics. So it's important now to step back and remember that bacteria are really different than the eukaryotes that we've been talking about. So in order to be complete in a genetics course, we really need to cover how prokaryotes' genetics work. Second, we're going to talk about them because they have been so very important in the study of eukaryotic genetics. We have used some of this stuff in lab already in the PGLOW project, and understanding the normal way that bacteria function is going to help us to understand these methods that we are using in molecular biology in our lab project. Bacteria, microorganisms are very, very different from eukaryotes. Generally, they're unicellular. They grow asexually, so that's just by binary fission. And the two daughter cells are going to be identical to the mother cell. Usually, bacteria have a single circular chromosome, and they don't have a nucleus. A few bacteria have multiple circular chromosomes. Bacteria grow very fast. Often we will grow them in a suspension, in a liquid suspension, or on an agar plate. And so if we spread bacteria on an agar plate, they are going to grow overnight into something that is visible. Individual cells that get spread out will actually grow into a whole clump of cells, and this is called a colony. And when you have a colony of bacteria, each of the cells in that clump are all identical to each other because they came from that single cell that you initially put on the plate. Now, bacteria do not, in fact, have sexual reproduction, but they do have several different ways that they share genetic information with each other, and this is going to allow recombination or a mixing up of the genetics. What we're going to focus on in this chapter is the three main ways in which bacteria exchange genetic material with each other. Couple of other things I need to introduce. Bacteria are prototrophic, and what that means is that they can make all of their own required nutrients. And so what we can do is put bacteria on a very simple plate, what we call minimal media, and this is just a sugar energy source, and they can grow. In contrast, you can also isolate strains of bacteria that are called auxotrophs. Now, auxotrophs have a mutation in one of the genes that is required for making some of their essential nutrients. So if they have a mutation, now they can't make their own nutrients anymore. So auxotrophs, we can get them to grow if we provide them that nutrient that they are missing. So adding amino acids, vitamins, whatever they are mutant for. So they can grow in complete media, but not on the minimal media, which does not include those extra things. A lot of bacteria have what are called plasmids, and so plasmids are very small circular pieces of DNA that are kind of extra chromosomal. So they are outside of the normal bacterial chromosome. They are a lot smaller than the normal chromosome. So they can be exchanged pretty easily between bacteria, and generally they can harbor a few genes on them. So plasmids are generally not considered part of the bacterial genome, but we can certainly use plasmids to provide some gene, for example, an antibiotic resistance gene, which will then allow that bacterium to grow on that antibiotic. Okay, in this chapter, we are going to focus on three main ways that bacteria can exchange DNA among themselves. Those three ways are conjugation, transformation, and transduction. This video will include only conjugation. To briefly summarize these three ways, conjugation uses a mating connection called a pilus to transfer a plasmid from one cell to another. Transformation allows a bacterial cell to take up free DNA from the environment, so maybe from a dead cell. And transduction uses viruses as an intermediary to transfer DNA from one cell to another cell. So first, let's talk about conjugation. I want to tell you about an experiment that was done by Lederberg and Tatum. This experiment was really the discovery of conjugation. So these folks took these two bacterial strains. One of those strains was an auxotroph for three different amino acids, threonine, leucine, and thi, so this particular strain could not make their own amino acids, these three types. Another strain was mutant for some other genes. So this particular strain on the right could not make its own biotin, phenylalanine, or cysteine. And so neither of these bacterial strains could grow on minimal media. And you can see that there is no colony when they're plated. It turns out that Lederberg and Tatum took these two bacterial strains, mixed them together, and then plated them on minimal media. And they were really surprised to see that there were some colonies that could grow. They were very few, but there were some that could grow. The reason they could grow is that there was some kind of genetic recombination or sharing of genetic material, so some of the cells had functioning copies of all 6 of these genes. Now, they didn't know at first what this meant, and one thing that they wondered is, do those bacteria secrete something that the other cell can use, or do individual cells actually change their genetics? So they did a modified experiment in which they mixed these two together, but kept them slightly separated with a filter. What they did is the media that strain A was growing in, they sucked it through to strain B, and they asked, can strain B grow if they get the media that came from strain A? And what they found is that no, they could not in fact grow. And so Lederberg and Tatum concluded that this genetic exchange between these bacteria actually requires physical contact between the bacterial cells. It's not just something that they secrete into the media. These experiments by Lederberg and Tatum were the first observation of conjugation between bacterial cells, and subsequent work by them and a number of other people showed that there is a plasmid called a fertility factor that is found in some bacterial cells. And so if a bacterium harbors the F plasmid or the fertility plasmid, it is able to make these projections called pili. Singular would be pilus. These projections can then contact a neighboring cell, and DNA can actually be transferred via the pilus from the bacterial cell that has the F plasmid to one that does not have an F plasmid. So to summarize how the F factor works, a bacterium that harbors an F plasmid, shown in red here, is called an F plus cell, and one that does not have an F plasmid is called an F minus cell. And this one with the F factor can form a pilus, and that pilus will then contact with an F minus cell, and once that contact is made, we can actually transfer the F factor from one cell to the next cell. And importantly, this process, conjugation, results in a copying of that F plasmid so that you started out with one F plasmid and you end up with both cells having an F plasmid. The way this actually works is one of the strands of DNA gets a little cut in it, and it starts to transfer that single strand over to the F minus cell. As soon as that single-stranded region gets transferred over, the region that is left single-stranded in the F plus cell immediately gets replicated to make it double-stranded, and also that new strand that's going over to the F minus cell becomes double-stranded. Now, as people learned more about how the F factor functioned, they realized that this mechanism was really only transferring the F factor. And so this should not be a mechanism whereby bacterial genes can be transferred from one bacterium to another. So additional work identified something called an HFR strain. And an HFR strain is actually a strain that started out as an F plus strain of bacteria, but it got these unusual characteristics. When an HFR strain was crossed to an F minus strain, you get many more recombinants that transferred bacterial genes from that F plus strain to the F minus strain. And so the HFR actually stands for high frequency of recombination. Secondly, when these HFR strains were crossed to F minus strains, those resulting cells did not actually usually become F plus. So it turned out that HFR strains derive from an F plus cell that has had the fertility factor insert into the genome. And so here we just have a depiction of how that could happen. You just get a little recombination, a nicking of the DNA, and they join together in the wrong way, and all of a sudden now you have the F factor inserted into the bacterial genome in blue here. So what happens here is that the HFR cell still has all of the genes to make a pilus, and in fact it tries to transfer itself into an F- cell once it conjugates with an F- cell. The problem is it doesn't realize that it can't do it properly anymore, right? So normally it would just transfer that little plasmid. Now what it tries to do is transfer itself, and in the process starts to transfer the entire genome of the bacteria. Okay, so here is how this works. So here is a HFR strain, and it will in fact conjugate to a cell that is F-. This F factor provides the information to make a pilus. That pilus connects with the F- cell, and it begins to try to transfer itself, thinking it's just a little plasmid. And instead, it starts to transfer the whole bacterial chromosome. And so here, we started it here, and we are going around, and we're transferring a portion of that bacterial chromosome. Eventually, the pilus will break up, and this portion of the bacterial chromosome is now called something called an X conjugate. And that X conjugate cannot be maintained in a bacterial cell on its own because it does not have all the characteristics of a chromosome. However, it can undergo recombination before it gets degraded, and if it does undergo recombination, any genes that were transferred from the HFR cell can actually be incorporated into the recipient cell. And so here you have this gene, it's the green gene. The black one here is maybe a mutant version of that same gene. And if you get this recombination, you can get replacement of the defective gene with a functioning gene. So this kind of transfer is actually what provides the explanation for what Lederberg and Tatum observed with their auxotrophic bacterial strains. I'm going to tell you about one other kind of bacterial strain that involves F factors. So sometimes we have HFR strain, and occasionally that HFR strain will actually pop back out of the bacterial chromosome and become a small plasmid again. When that happens, it can come out perfectly so that just the F factor is now separate from the chromosome, and it would just become a normal F plus strain again. Or alternatively, sometimes there's a mistake that is made and a neighboring gene from the chromosome can actually go along with it mistakenly. And so this purple part here mistakenly got included in the F plasmid, and now this plasmid is not just the standard F factor, it is now called an F prime cell because it actually carries one or several genes from the bacterial genome. When this gets conjugated into an F- cell, you can end up with a cell that is called a partial diploid. Normally in bacteria, we just have one of each gene. In this particular cell, there's two of that purple gene, one that is harbored on the bacterial chromosome, the other one that got put onto that F plasmid. Now, going on in time, there are another couple of people, Jakob and Wolman, were doing some experiments with conjugation with HFR strains, and they realized something really cool, and that is that we could actually use this conjugation to make bacterial chromosome maps. So if you start to transfer that F plasmid, you're going to start to transfer that whole chromosome. After a certain period of time, you're going to transfer the first gene that you get to in that bacterial chromosome, so maybe this purple gene, and then after a little bit more time, you'll transfer the blue gene, right? If you wait a little bit longer, eventually you can get that orange gene transferred, and finally possibly the green gene. So Jakob and Wolman mixed these HFR cells together with F- cells for different periods of time and then allowed that mating and then put them in a blender to separate and break up that pilus. And what they measured then is how long did it take to get recombinants in which that particular gene had entered the neighboring cell. So here's an example of their data. So this is looking at 4 different genes. And so what they found is, so about 10 minutes after mixing them together, the first azi recombinants were observed. Just a few minutes later, the first ton recombinants, and then toward 18 minutes, the first lac recombinants, etc. And what you see here is you can actually make this map and put all of these things on that map with the units in minutes. So other experiments indicated for the very first time ever that the bacterial chromosome is circular. So people were using different strains that were different insertion sites for the HFR. And so some strains had an insertion at one spot, another strain might have an insertion at a different spot. And these different lines would actually give different chromosomal maps. And so here's the F plasmid. The first one to transfer is Lu, then 3, then Thi, etc. In this other line, the first gene to transfer was Thi, then 3, then Lu, then azi. And this actually showed the Lu and the azi genes next to each other, whereas the map from the other line showed the Lu and the azi genes on far ends of the map. And so people first got the idea from these experiments that the bacterial chromosome is probably going to be circular. Subsequent work has made this much more complicated bacterial chromosome map, but I just want to point out that the units are minutes, right? So here are the actual units, and it is possible on really rare occasions to transfer that entire bacterial chromosome, and it takes about 90 minutes to transfer it. So in summary, in this particular video, we looked at conjugation. And there are 2 main things that can happen with an F plasmid. This cell that is making a pili will connect to an F- cell and transfer that F plasmid that is separate from the bacterial chromosome. And the result of that would be 2 cells that are now both F+. In contrast, if that F plasmid inserts into the bacterial chromosome, it becomes an HFR chromosome or an HFR strain, and it will try to transfer the entire bacterial chromosome from the HFR line to an F- strain. Usually it does not manage to go all the way around, and so most of the time you're going to end up with a bacterial cell in which there are some recombinants, so some bacterial genes got transferred, but usually the entire F plasmid does not get transferred, and the new cell won't actually be able to make a pilus on its own.